Recall the 5 conditions of Hardy-Weinberg. 1. Chance events do not alter allele or genotype frequencies. This is more likely to be the case in very large populations. 2. Mating between individuals is random with respect to the trait in question. 3. Mutation does not change allele frequencies. That is, the chances that any new allele will be created by mutation equal the chances that a mutant allele will mutate back to the older form. 4. There is no gene flow. Individuals do not migrate out of the population, and new individuals do not migrate into the population. 5. There is no selection acting on the individuals in the population, so each genotype has an equal chance of surviving and reproducing. As we go through lab, keep these conditions in mind. Evolution is defined as any change in genotype or allele frequencies across generation. But can we observe such changes? In our case, we will be using fruit flies to help us understand evolution. To observe evolution, we will compare the expected genotypic frequencies under Hardy-Weinberg conditions with the observed genotypic frequencies in a later generation. The fruit flies we use have genetically engineered eye colors that are incomplete dominant. This allows us to discriminate between flies with 3 genotypes: C1C1 red eyes, C1C2 pink eyes, and C2C2 white eyes. In the lab, we took 12 samples of flies identified as S0 or S1. We sorted the flies from each of these sample vials. Since the eye color in males can be harder to distinguish, the flies must first be sexed. Male fruit flies have sex combs and their abdomens end in a ring of small bumps. In females, there are no sex combs, and the abdomen ends in a pointed structure. After being sexed, the females were divided based on eye color. The data from the whole set of vials was then compiled. Now recall that the vials were labeled either S0 or S1. In the lab, we have raised 3 populations of fruit flies. The first is a large source population of fruit flies, and the second is a small founder population. The source population lives in the laboratory, passing from one generation to the next as individuals mate, reproduce, and die. The source population has flies representing all 3 genotypes. For the first part of the lab, half the data is for the source population at the first generation, S0, and the other half of the data is for the source population at the second generation, S1. The S1 samples were taken from the same source population, 1 generation later. You will use the genotype frequencies from the source population at T0 to calculate the allele frequency at T0. From this, you will calculate expected genotypic frequencies. You will compare the expected genotypic frequency from the S1 population at T1 with what you observe at T1 to see if evolution is occurring. For the second part of this exercise, we will analyze the genotypic and allelic frequencies in a small founder population. A mated female was put into the founder vial, which means the genetic diversity of her population was very low. The female carried the sperm of 1 or 2 males with whom she mated before she was removed from the source population. At its inception, the founder population contained the genes of only 2 to 3 individuals. However, because each female subsequently produced many offspring, each founder vial now has a relatively large population in the F1 generation. We have provided the data for the S0, S1, and F1 populations for you. You will be responsible for completing the calculations for the S1 and F1 populations. Now, Dr. Pat Randolph will show you how to do the calculations using the S0 population.
Okay, so in the lab manual, they describe the experiment we're doing with population genetics with fruit flies. But if you recall, we're going to have 3 populations we're looking at effectively. We're going to have what we call our S0, which is our source population. It's a large population of flies that we maintain. Then we pull the generation out of that, we call the S1. It's also a very large population of flies growing at the same conditions and the same environment as the S0. Then we have what we call our F1 generation of flies or population. And this was produced by pulling just a few individuals out of the S0, kept in isolation from these other populations, and allowed to grow larger on their own. So the questions we're asking is, for this particular part of the lab, is the S1 population evolving with respect to S0? And is the F1 evolving with respect to S0? You could probably make some predictions on that based on just what I told you. And so this table here, which will be in the lab manual for you are the data from a previous quarter that were collected. So here we have the different populations, S0, S1, and F1. Here we have the eye colors here, red eyes, pink eyes, white eyes. And then here we have the number of females that were counted from the different groups and pooled together. I'm going to focus on the S0 population here, the red, pink, and white for the S0. If I add these together, we get a total of 202 individuals. And that's important for us when we start thinking about genotype frequency calculations. Okay, let's calculate some genotype frequencies. And again, I'm going to focus on the S0 flies. So I've rewritten this table here just to make things a little bit more clear. Again, remember we're focusing on eye color, fruit fly eye color. There's a single gene with two alleles, a C1 and a C2 allele. So we had some flies with red eyes, some with pink, some with white. And if you recall from the lab manual, these alleles are incompletely dominant. And so the pink eyes here are the heterozygote. They have one copy of the C1, one copy of the C2 allele. The red eyes are homozygous for the C1 allele, and the white eyes are homozygous for the C2 allele. So we have the counts up here. So we have 50 red, 120 pink, 32 white. I want to convert that to a genotype frequency, or what proportion of the flies are the C1C1 genotype, what proportion are C1C2, what proportion are C2C2. So to do that, what we're going to do is, for the red eyes, we saw we had 50 individuals. I'm just going to write this here. And then we're going to divide that by the total number of flies for that population. And that's where the 202 comes into place here. So we had 202 individuals. Divide 50 by 202. That'll give us the genotype frequency for this particular group of flies. So you do the math, it's going to be a value of 0.248. Or about 25% of the flies from the S0 have red eyes, or that C1C1 genotype. So we want to do the same thing for the C1C2. So go back to my data here. We had 120 pink flies. So 120 divided by 220. That'll give me a value of 0.594. So again, about 60% of the flies are pink, which makes sense. We had 120 out of 202. It's a pretty large proportion, about 60%. Then we do the same thing for C2. We had 32 flies. Take 32, divide that by 202. And that'll give us a value of 0.158, or about 16%. Just a quick note, you only need to go 3 decimals. Don't get crazy with decimals. 3 is plenty for us. Okay, so these are our genotype frequencies. What proportion the C1C1 occurs, what proportion of the C1C2 occur, and what proportion of the C2C2 occur within our S0 population. Okay, what do we want to do with that? So we want to, out of these, calculate the allele frequencies. So basically thinking about how many times does the C1 allele occur within this population? How many times does the C2 allele occur within this population? So how do we go about doing that? And so the pre-lab went through this process with you. The lab manual goes through this process with you, and I'm going to go through this process with you right now. So again, all we're doing is counting. All we're doing is counting how many times does the C1 allele occur and how many times does the C2 allele occur. And so to do that, first of all, we're thinking about the C1 allele. We know the C1 occurs in the C1C1 homozygote. So we want to take that genotype into account. We also know the C1 allele occurs in the heterozygote, the C1C2. So we want to take that genotype into account. But since we're just counting, we want to add these two together. And this just represents the frequency. C1C1 is going to be the frequency of the genotype. C1C2 is going to be the frequency of that genotype. But what we also want to do then is, for the heterozygote, multiply that value by 1/2. And the reason we do that is, in the heterozygote, only half the alleles are the allele we're interested in this case, C1. And so that's why we're going to multiply that by 1/2. Then all we need to do is just take the values that we got. So the C1C1 genotype, we calculated 0.248. I'm just going to put that number there. We add to that 1/2. the frequency of the C1, C2, which is 0.594. We do that math, and we get a value of 0.545. So about 55% of the alleles within this population are the C1. Then we can do the same thing for C2. So we know we're interested in the C2 as a homozygote, and that C2 allele also occurs in the heterozygote. Do the exact same calculation, but we're just going to use the values for the C2, C2 genotype, so that's 0.158. Add to that the heterozygote, 0.594, same value that we did up here. And you do that math, you get a value of 0.455. So about 46% of the flies have that C2 allele within that population, or at least the C2 allele occurs at a proportion of 46% within that population. And just a quick math check, we look at these should add up to 1. And looking at that eyeball on it, yeah, that adds up to 1. That tells us I didn't make any math errors. So now what do we do with that? So we calculated genotype frequencies, we've calculated allele frequencies, so what? What are we going to do with that? Now we want to make predictions. So this is where we get to the Hardy-Weinberg equilibrium. Hardy-Weinberg allows us to make predictions for a population what those genotype frequencies should be if that population is not evolving. And so to do that, we needed the allele frequencies. And so to figure out what those frequencies should be, we can use another tool we call the Prout Square. And so the Prout Square, if you look at it, I just built this here. If you look at it, you didn't see that. If you look at it, it looks a lot like a Punnett square you did with Mendelian genetics. But now we're thinking about the level of the population. We're in a Punnett square, you'd had one parent here, another parent here. Think about the product in the offspring. With the Prout square, we're interested in the population level. So effectively, again, we're seeing contributions from one set of parents, contributions from another set of parents in the offspring, but think about this again at the population level. And so in this case here, the C1 allele occurred at a frequency of 0.545, so I'm just going to put that number up there. That just came from our calculation we just did. I'm going to put the same value here, 0.545. Then the C2 allele also came from our calculation here, that's 0.455. And then C2 here is also going to be 0.455. Okay. Now, just like a Punnett square, we want to think about, okay, what's the probability if one parent in the population or the males in the population have a C1 allele, which occurs about 55% in that population, and then the females, that allele also occurs about 55%. What's the probability of those sperm and eggs are going to come together, given that alleles occur at those proportions? And so, again, we're thinking about this genotype here, the C1 genotype. And to figure that out, we just multiply this frequency times that frequency, the 0.545 times 0.545, or 0.545 squared, however you want to think about it. And that answer, sorry, I gotta grab my cheat sheet over here. And that answer is going to be 0.297. So we would do the same thing for the C2 homozygotes. So say the males are going to contribute C2, the females in the population are also going to contribute C2. What's the probability that sperm and egg carrying those alleles are going to come together given the alleles are occurring at these proportions? Same idea there. Got C2, that's going to be 0.455 times 0.455. And that's going to give you a value of 0.207. Then we can think about the heterozygotes. There's two ways to get heterozygotes. The males could contribute the C1 and the females the C2. That's one possibility. Or the male could contribute the C2 and the female the C1 in that case, to also get a heterozygote. So there's two ways to get that. But we do the math the same way. So I just take this allele, multiply by that. So that's 0.545 times 0.455. Then I do the same thing here, 0.455 times 0.545. And then to figure out the heterozygotes, I would add those two together. because there's two ways to get the heterozygotes. Once I've done the multiplication, I add those two together, and I'll get a value of 0.496. And so that would be the expected frequencies of the heterozygotes. So we can write this a little more cleanly. So we expect the homozygotes to occur at a frequency of 0.297. The C2C2 homozygote at a frequency of 0.207. Then the heterozygotes to occur at a frequency of 0.496. Keep in mind, this is the same thing as p squared. This calculation is the same thing as q squared. Adding these two together is the same calculation as 2pq within that Hardy-Weinberg expression. So however you want to do it is fine. Some people like to do it a tabular way like this. I like it. I'm a visual person. It works for me. If you're comfortable with p squared, q squared, 2pq, that's perfectly okay. Okay, so again, these are our expected frequencies for the S1 population. So what you're going to do as part of the homework then is go back to our S1 over here, calculate the genotype frequencies, what we call the observed, what's actually there. You're going to compare those to the S1, these frequencies here, then decide are they the same or are they different. And we're using a threshold of a value greater than 0.05. So we're not going to run statistics on this. We're not going to do that to you this week. So if it's a difference of greater than 0.05, we're going to call it different enough. Then you can do the same thing for F1. You're going to calculate expected for the F1. Remember, the F1 originate from the S0. So we're comparing the F1 with the S0. Calculate the expected for the F1. Then you're going to calculate what we actually see here. Again, make the same comparison you do with the S1 and decide, is F1 being maintained at Hardy-Weinberg or has it also evolved? So it does take some practice to work through these. You want to be comfortable with these calculations. They will show up on the lab exam, and quite often similar calculations will show up on lecture exams. The math's not hard. It's just a matter of getting the numbers in the right place. If you have questions about how to do this, by all means feel free to contact your TA. We want you to be successful at this.
That's it for this video. Remember, if you have trouble with any of these concepts, please contact your TA. Thank you for watching.